has several kind of weathers.
Yeah.
Yeah. Yeah, it's comparing with Charlottesville is, you know. Yeah. Okay. Maybe we can start now.
Okay.
I'm gonna do a presentation. Okay. Good morning, everybody. Thanks for coming to our institutional seminar this Friday. I have the honor to introduce you to Dr. Mark Benhacker. Benhacker or Benhacker? How do you pronounce it?
I say Bean Hawker. I've heard many different variations.
Yeah, I suppose. Yeah.
They're all good.
Yeah, thank you. Dr. Mark Benhacker is a researcher at the Department of Pharmacology in the University of Virginia in Charlottesville. And actually, Dr. Benhacker is a neuroscientist to the bone, because he has his bachelor's degree in neuroscience at the Washington and Lee University in Virginia, and then he had two summer research courses at the Woods Hole in Massachusetts. One of them is about neural systems and behavior. The second one was about methods in computational neuroscience. And then he got his PhD degree at the neuroscience also at the University of Pennsylvania in Philadelphia. And then he went to perform his postdoctoral training at the John Hugenar's lab at Stanford University. Dr. Hugenar is actually a pioneer in the researching of neuronal mechanisms that underlie synchronous oscillatory activity in the thalamus, the cortex, and the massively interconnected thalamocortical system. This was actually like the cherry on the cake for Mark, I suppose, in his training. And then he got his academic appointment at the University of Virginia since 2011 as an assistant professor, and lately in 2018 as an associate professor. Mark has several honors and awards. In 2004, he got the Saul Vinegran MD Award for the best dissertation in neuroscience at the University of Pennsylvania. Then in 2007, he got the Eric W. Lotman Training Fellow and Honorary Epilepsy Foundation Fellowship. And in 2015, he got the Peter C. Isaacson Mentoring Award at the Department of Pharmacology. And Mark has so far around 32 publications, very interesting publications about epilepsy, the mechanism of epilepsy, the relationship with the ion channels. And I want to acknowledge and to thank a lot Mark for accepting this invitation to present us this seminar. And I think this could be, in this case, could be a very good possibility to start some more collaborations between our labs and our institutes. So I really appreciate your availability to present us this seminar, Mark. So please, thank you.
Yeah, thank you for that very kind introduction, and thanks for having me. I wish it wasn't through the lens of a computer camera.
Yeah.
But maybe one day I can come visit.
Sure.
It sounds like a wonderful spot for sure.
Yeah, hopefully.
So I'm going to share my screen. Can you all see that?
Yes, that's good.
Okay.
Yeah, perfect.
It's good. Okay. Yes. So thanks again for that wonderful introduction. And you can hear me fine?
Yes.
Okay, good.
Yes.
So my lab studies the neural circuits that drive seizures associated with epilepsy. And today I'm going to talk about a couple of phenomena that we've known about for almost 100 years. And these are phenomena that clinicians know well. And these phenomena relate to how environmental conditions can provoke seizures. The work that I'll be describing today is not yet published. These papers are in review. I hope soon they will be published. I'm waiting. So by way of introduction, broadly speaking, there are two forms of epilepsy, focal and generalized epilepsy. And in the focal forms of epilepsy, the seizure begins in a relatively restricted portion of the brain and is restricted there. Whereas in the generalized epilepsies, the seizure begins in a restricted spot, but then rapidly invades most parts of the brain. Collectively speaking, there are roughly 50 different forms of epilepsy, and they all differ in terms of their structures involved, the age and sex of the individual, whether genetics or trauma are driving the seizures. The idiopathic generalized epilepsies, which are now actually generally referred to as the genetic generalized epilepsies, comprise about 65 to 70% of all epilepsies. And within this class of idiopathic generalized epilepsies, we find a few subclasses: juvenile myoclonic epilepsy, generalized tonic-clonic seizures, and childhood and juvenile absence epilepsy. And my lab focuses on these latter two epilepsies, the absence epilepsies. Electrographically, these seizures associated with absence epilepsy have this very stereotypical spike and wave discharge form. And in the last few decades, we have learned that several mutations, including loss of function mutations in the GABA A receptor or in the CLC-N2 chloride channel, or gain of function mutations in T-type calcium channels, can all contribute to absence epilepsy. However, the penetrance of any of these mutations is pretty low in the absence population. So unfortunately, it's very difficult to draw a straight line between any one of these mutations and absence epilepsy. In addition to the genetic component of these seizures, we've also learned that the environment or external factors can modulate the occurrence of these seizures. So for example, we've learned now almost 100 years ago that hyperventilation, an increase in respiratory rate, can reliably provoke these seizures. And we've also learned that diet, namely blood glucose levels, can also modulate the occurrence of these seizures. And my lab has really focused in the last few years on how these factors can drive the occurrence of these absence seizures. So I'd like to provide a little bit of historical perspective for this. So as some of you may know, in 1924, Hans Berger was credited for first developing human EEG recordings, and he did this mostly for alpha rhythms in healthy humans. Unfortunately, his novel technique at the time was generally unnoticed in his home country of Germany, but it didn't go unnoticed by British EEG, by British colleagues, Edgar Adrian and Brian Matthews. And then shortly after they discovered it, it was also discovered by American epileptologists William Lennox and Frederick and Erna Gibbs. And they really were able to apply the EEG to epilepsy. So what I'm showing you here is one of the very first recordings in 3 patients here, EEG recordings of a spiking wave discharge during an absence seizure, which is also sometimes referred to as petit mal seizures. And I think with these recordings, you can really appreciate, because of the filtering properties of these electrodes, why we call these a spike and wave discharge. There's a very noticeable wave and a very noticeable spike to this EEG. So after they were able to capture seizures with the EEG, the same group then, just a few years later, discovered that glucose levels, specifically low blood glucose levels, hypoglycemia, can provoke absence seizures. And then just a year later, they published a paper showing how hyperventilation can very reliably trigger absence seizures. So to put this in a little more modern perspective, ethosuximide, which is still one of the main drugs to treat absence epilepsy, came on board in 1960, followed by valproate in 1967, and then lamotrigine in 1994. And these three drugs, despite their old age, are still the three primary drugs used to treat absence epilepsy. Notably, lamotrigine is the least effective, which was a conclusion recently made by Tracy Glouser's group at Cincinnati Children's Hospital. So today, I'm going to tell you two stories that sort of go back to these discoveries that were made many, many years ago. First, that hypoglycemia can trigger seizures, as well as hyperventilation. So I'm first going to talk about hypoglycemia. And this is the original paper published in 1939 and entitled Influence of the Blood Sugar Level on the Wave and Spike Formation in Petit Mal Epilepsy, again, also known as absence epilepsy. And even though we have known about this phenomenon for such a long time, we still have virtually no idea how this occurs. However, more recent studies have supported the hypothesis that absence seizures and blood glucose are linked. So first, about 10 years ago, the big epilepsy group in Australia discovered that GLUT1 mutations can account for a significant portion of early onset childhood absence epilepsy. So GLUT1, as you know, is the primary glucose transporter that shuttles glucose across the blood-brain barrier. We've also learned that in healthy humans, this was done in PET scans, healthy humans, hypoglycemia, even just a little bit of hypoglycemia, can increase thalamic activity. And this is, of course, relevant, as I'll get into later, because the thalamus has long been considered to be important for the generation of absence seizures. And then we've also observed that hypoglycemia can provoke absence seizures in rodents. So first, in 2010, in the DBA2J mouse, and then as I'll show you on my next slide, also in this WAG rye rat, which is a long-standing model of absence epilepsy. So this is the experiment that we do to evaluate this. So in panel A, you can see the general experimental approach. So we record seizures in both of these animals. These are spontaneous seizures. So these animals have seizures just spontaneously. We don't have to do anything to provoke them. They have a spontaneous seizure level at basal conditions, including in the fed state. But then what we do is we withdraw the food for about 12 hours or so, and then we record the EEG again. This is an example of what a spike wave seizure looks like in one of the rats. You can see that it's a generalized event insofar that you can measure it on both cortical hemispheres. It's also associated with behavioral arrest. You can see the loss of muscle activity, also a hallmark of absence epilepsy. And during the seizure, it's enriched in this 3 to 7 hertz range, which is stereotypical of these spike and wave discharges. This right here is just an expanded view of this, where you can really see the spike and wave nature. So in panel C, I'm showing you raster plots. So each of these dots here represents a seizure. In the mice, so we recorded 10 mice here and 6 rats here. And this control condition corresponds to when the animals are fed. You can see that at baseline levels, the level of seizures is higher in the rats. This is well documented. However, in both rodent models, if we fast the animals and record seizures again, we can see an increase in the seizure count, which is quantified here for the mice. So the y-axis here is the number of spike wave seizures recorded, And you can see there's an increase in the fasted condition relative to the fed condition. As you can also see in the schematic, we draw blood from these animals so we can confirm that they're indeed hypoglycemic. You can see that in the fasted animals, not surprisingly, their blood glucose levels are lower. Also, as you may know, during fasting periods, you undergo ketosis and you have an elevation in beta hydroxybutyrate, one of the ketone bodies. So this elevation in ketone bodies in the serum is also not surprising. This is well documented. And in the WAG rat down here, we get the same general conclusion. Fasting increases seizures, we get a drop in glucose, and we get an increase in ketone bodies. So considering that we got both a drop in glucose and an increase in ketone bodies, we were hoping that we could sort of dissociate these two to see which one of these was most important for the increase in seizure count. So the way we did this was by using insulin, which is a well-known way to drop sugar levels. We also hypothesized that at least acutely, an insulin injection would leave ketone bodies the same. And that turned out to be the case, as I'll show you in a moment. So the experiment was that we record seizures in the baseline condition, and then we inject animals with either saline or insulin. We also collect blood to confirm that, that we're dropping glucose, and we're also measuring ketone bodies. And just as with the fasting, the insulin injection, just an acute single injection, increases the seizure count. I'm only showing you the rat data from now on. The mouse data are the same. And as we predicted, the injection of insulin produced a significant drop in blood glucose. So here, this is before and after the injection of saline. So we collect blood both before and right here during the injection. And not surprisingly, saline does not change blood glucose levels. However, insulin does. And then the beta hydroxybutyrate, the ketone body, was unaffected by insulin. So this to us suggested that it is indeed the hypoglycemia that is triggering or provoking these absence seizures in the rats. So at this point, we have some phenomenology, but we wanted to understand the mechanism. So there's a very rich literature, as Juan Carlos alluded to, that has established that it is circuit connections between the cortex and the thalamus that really underlie absence seizures. And this dates back all the way to the 1940s. So here you see the primary players in the generation of absence seizures. So you have the cortical, you have the cortex, and then you have the dorsal thalamus, which is the structure that contains the primary sensory nuclei, as well as higher order thalamic nuclei. And then on top of the dorsal thalamus, you see this shell of inhibitory neurons that make up the reticular thalamus. So the circuit that is sort of the canonical circuit to describe absence seizures goes as follows. A bout of hypersynchronous activity in the cortex is then relayed to the thalamus. Namely, it activates the reticular thalamus, again, this inhibitory GABAergic structure. These GABAergic neurons then provide feed-forward inhibition to thalamocortical neurons in the dorsal thalamus. These neurons, as I'll describe in more detail in a moment, are enriched in low-threshold T-type calcium channels that promote the expression of post-inhibitory rebound bursts. So following inhibition, these neurons produce a burst of activity, paradoxically. This was first described by Linas back in 1981 and 82, I think. And that burst of activity then is relayed back to the cortex as well as to the reticular thalamus. These are excitatory connections. And it is this back and forth between feed-forward inhibition and recurrent excitation that drives an oscillation between these two structures. And because these are thalamocortical neurons, that oscillatory activity is imposed on the cortex, which is why we can record these seizures cortically through the EEG. Okay, so this is the canonical circuit describing how absence seizures are generated. So considering this rich literature implicating the thalamus in the generation of these seizures, we next wanted to see if we could selectively target glycolysis in the thalamus and record seizures. So the experiment went as follows, where we cannulated our rats. So we delivered a cannula into the dorsal thalamus, and then we delivered either saline or 2-deoxyglucose to disrupt glycolysis selectively in the thalamus. Oops. And as you can see, I already showed you, the seizure counts increase significantly. I was going to show you this panel first, which is sort of the time course of the experiment. So here, this is the saline condition. Here, this is just a stacked histogram showing you the contribution of spike wave seizure count to the entire population. We turn the pump on here. You can see that the rate of spontaneous seizures is unaffected by saline. However, if you deliver 2DG to the dorsal thalamus, you get this bump in seizures. So inhibiting glycolysis is sufficient to provoke these spike and wave seizures, as is quantified here. So glycolysis, of course, is a complex biochemical cascade that breaks down glucose to make ATP and ultimately pyruvate. So here I'm going to show you two key actions associated with glycolysis that can also regulate neuronal activity. So we have the activation of AMP activated kinase, as well as the activation of ATP sensitive potassium channels, KATP channels. So during hypoglycemia, reduction in glucose, ATP is reduced, of course, and you get a buildup of ADP. And this ADP then activates this AMP kinase, AMPK. In addition to that, because ATP is depleted and ATP blocks the KATP channel, this reduction in ATP alleviates the blockade on the KATP channel, and therefore activates this channel. And this channel then supports a more hyperpolarized memory potential in the neurons that express the channel. Okay, so I'm going to describe a hypothesis that first describing the potential involvement of KATP channels, and then I'm going to focus our attention on AMPK. So the hypothesis that we originally set out to test, because it seemed fairly straightforward, was that, as I described, a reduction in ATP during fasting would activate KATP channels and be in thalamocortical neurons, going back to our circuit. And because hyperpolarization of these neurons promotes rebound bursting in these neurons, and because rebound bursting appears critical for the generation of seizures, we thought that this activation of KATP activity would, in turn, ultimately increase the occurrence of these seizures. I'm not going to drag you through a lot of data that a very talented graduate student accumulated to test this hypothesis, because ultimately, even though we could demonstrate that KATP channels were enriched in thalamocortical neurons, we were not able to draw a line between KATP channel activity and hypoglycemia activated channels. So for that reason, I am now going to turn our attention to AMPK. Again, AMPK is activated during hypoglycemia. And the reason why we found this interesting is because this grad student in my lab observed or went back into the literature and found this curious result. So this is from Stephen Moss's lab, and he looked at dissociated hippocampal cells, and he recorded them with patch clamp electrodes, and he applied baclofen, which is a GABA B receptor agonist, okay? And what he observed is that if you repeatedly apply baclofen to these neurons, you get this nice outward current, which is driven by the activation of potassium channels. So postsynaptic GABA B receptors activate GERK channels, potassium channels. you get this nice activation of the current.
Excuse me, Mark. Sorry. Yeah. Sorry to interrupt you. Are you using a laser pointer on the screen or something? Because I mean, I think most of us, we cannot see any pointer, but I don't know. Okay. Actually, are you using it?
Can you see that?
Yeah, that one.
Okay, sorry.
Yeah. Yep. Sorry, no, sorry to interrupt you. Thank you.
No worries. Sorry about that.
Yeah.
Okay, so the experiment was as follows, again, from Stephen Moss's lab: record from dissociated hippocampal cells, apply baclofen, the GABA B receptor agonist. And what he observed is that with repeated baclofen application, you get this smaller response over time. Okay, and he called this a response rundown. However, if he included agents that activate AMPK within the recording pipette, he could augment this response and blunt the rundown. So that was shown here in an example form, but shown here in the population. So on the y-axis, we are looking at the K current, the potassium current, that is activated by postsynaptic GABA B receptors with the agonist baclofen. And you can see in red, in control conditions, you see this rundown of the current, as I described here. However, if you include AMP, which in turn activates AMPK, you get a reduction in this rundown. He also showed that another AMPK activator, metformin, which is also, of course, a common diabetes drug, can also blunt this effect. So the conclusion from this study was that activating AMPK can augment GABA B receptor activity. And the reason why we found this interesting is because there is also a historical link between GABA B receptor activity and absence seizures. So in rodent models, including the WAG, if you inject GABA B receptor blockers, for example, CGP, you can reduce the number of seizures. So this is on the y-axis, this is the duration that animals are seizing. In control, or you see the black circles, and in here, you see a reduction in seizure count in a dose-dependent manner when you apply the CGP blocker. Interestingly, the inverse of this result is also true. So baclofen, again, the GABA B receptor agonist, can increase seizures in a dose-dependent manner. So showing us that there is a link between GABA B receptor activity and spike and wave seizures. You can also find this link in a reduced thalamic slice preparation. So the circuit that I described earlier, you can actually retain in an acute thalamic slice and then do electrophysiology on that. And again, I want to highlight the importance of this post-inhibitory rebound burst in sustaining epileptiform activity. So again, feed-forward inhibition inhibits these thalamocortical neurons that are enriched in T-type calcium channels. And this inhibition then produces this post-inhibitory rebound burst that is then relayed back to the cortex and also sustains the oscillation here. So in this reduced slice preparation, you can record activity that is normal, not pathological. And that comes in the form of these sleep spindles, which are shown here in EEG form. You can also generate pathological activity, the spiking wave discharge. So these are the correlates of those two respective activities in a brain slice. So in these preparations, you stimulate the cortex and you evoke spindle-like activity. If you really stimulate the cortex very hard, you can generate seizure-like activity. So this is a correlate for absence seizures in a brain slice preparation. Now, what Terry Ball showed many years ago is that you can transform epileptiform activity into that normal spindle-like activity by including a GABA B receptor blocker. So we're here on the y-axis, we're just measuring the frequency of the evoked oscillation. And you can see that it's generally fairly fast, around 7 hertz. If you really stimulate the cortical neurons, you can transform the same slice into one that produces epileptiform oscillations. And then if you do that with applying this GABA B receptor blocker, the slice goes back to the normal one. Again, supporting the idea that GABA B receptor activity is important for the generation of this epileptiform activity. And this this results from this cooperative activity between low threshold T-type calcium channels and this post-inhibitory rebound burst that I will just point you towards this paper that a student of mine recently published, where he looked at computationally the relationship between GABA B receptors, T channels, and bursting. He does this in incredible detail. I'll spare you the details, but to be clear, there is a clear link between the temporal dynamics of inhibition and the probability that these thalamocortical neurons generate a rebound burst. And that's, again, can be found here. So I'm just going to distill what Adam found in a very simple cartoon, which is shown here. So what I'm representing here is the memory potential of a thalamocortical neuron. And here's depolarized memory potentials, here's hyperpolarized memory potentials. And these neurons generally their resting memory potential is around, let's say, minus 65 millivolts. Now, at this memory potential, these low threshold T-type calcium channels are closed, but they are ready to open. In other words, they're primed to open. And if there is a small depolarizing change in the memory potential, these channels will open. And they will permit the influx of calcium, which then drives this low threshold calcium burst, on top of which you see action potentials. Because these open channels are closed with about tens of milliseconds, the memory potential depolarization occurs for about that duration. So then the channels close. Their closure is time dependent. And in this state, the low threshold calcium channel is closed, but it's also important to note that it is inactivated. And an inactivated low threshold T-type calcium channel cannot open again upon depolarization alone. Instead, the channel has to be, the memory potential has to be hyperpolarized. This removes inactivation or deactivates the channel. And when that occurs, this channel is then, again, ready to be opened, primed. So GABA B receptor inhibition, and again, these thalamocortical neurons are enriched in postsynaptic GABA B receptors. They sustain a long lasting inhibition in these thalamocortical neurons, sufficient to deactivate low threshold calcium channels that then enable those neurons to generate a calcium spike that produces a rebound burst. Okay, so that's generally what Adam was modeling in his paper. So, so that brings us back to this AMPK modulation of GABA B receptor activity that I described from Steve Moss's lab. So a graduate student in my lab, Katie Salvati, wanted to first see if you could activate AMPK in the thalamus. So she did this in using Western blots in acute thalamic slices. And she measured activated or phosphorylated AMPK. And relative to control solutions, if she applied for about 30 minutes potent AMPK activator, A7, metformin, or 2DG, she could get an increase in activated AMPK. She also used a fret indicator. So this was done in real time now, where we with actually Ed's help, we delivered this AMPK reporter called AMPCAR so that it would be expressed in thalamocortical neurons. And then we recorded the fret efficiency, and both the potent A7 AMPK activator and metformin increased fret efficiency, indicative of activated AMPK. So in the thalamus, you can activate AMPK. And this is just quantified right here. So then, just like Steve Moss did, Katie wanted to see if she could augment postsynaptic GABA B receptor responses evoked by baclofen by including AMPK activators in the pipette. And lo and behold, she could do so. And she did this in an acute thalamic slice, as you can see here. This is a baclofen puff with just control pipette solution. And that control solution is represented in all of these black dots, similar to what Steve Moss found. We see a gradual rundown in the response. However, if she included the AMPK activator, either AMP, metformin, or this potent A drug, she was able to augment the postsynaptic GABA B receptor response. Again, the postsynaptic GABA B receptor is critical for sustaining the post-inhibitory rebound burst in these neurons. So she then turned to an acute thalamic slice that I already described, one that can produce network oscillations. And she evaluated whether these AMPK activators could also augment this epileptiform activity recorded in this acute slice. So she evokes these oscillations, and this is the control condition, and then applies metformin. And she observed that there was a prolongation of the epileptiform activity. This is shown for a whole experiment in time series. So this is like a pseudo raster plot. Each of the waves indicates an oscillation. This is the first stimulation in the experiment. This is the last stimulation in the experiment that lasts about an hour. And then as she applied metformin, you can see that the oscillation increased in intensity. And this is quantified here for metformin. She also did the same thing with this other potent AMPK activator, A7. And critically, she showed that if you first incubate the slice in a GABA B receptor blocker and then activate AMPK with A7, you get no change in the resultant evoked activity, suggesting that this prolongation is indeed GABA B receptor dependent. And this is quantified here. So we can augment postsynaptic GABA B responses with AMPK activation. We can intensify evoked epileptiform oscillations. So then she wanted to see if she could also increase seizure count in vivo. So again, she cannulated animals as before, and then compared the delivery of a vehicle saline solution to this AMPK activator. And just as before, the AMPK activator, again, just in the dorsal thalamus, was sufficient to increase the seizure count. And there was no change in seizure duration. We also measure that. It's not particularly relevant here. But this showed that AMPK activation was sufficient to increase seizure count. So the model that we're proposing now is that for hypoglycemia evoked seizures is that it is the aggravation of thalamocortical circuits that drive this hypoglycemic response. So during a drop in glucose, even acutely, you get an elevation in a phospho-AMPK. This is the activated form of AMPK, and that augments inhibition mediated by GABA B receptors in thalamocortical neurons. So it makes the inhibition more robust, which as a consequence makes rebound bursting more robust in those thalamocortical neurons. And when rebound bursting is more robust, you get an increase in the seizures. I should point out that ethosuximide, that drug that I described in that timeline that is used to treat absence epilepsy, is a potent blocker of rebound bursting in thalamocortical neurons. So that is our model for how hypoglycemia provokes these absence seizures. And just to conclude this part, the way we think about this now is that the brain, as the brain goes through fed and fasted states, it will go through states that are either
a question.
Yeah,
please?
Sure.
Ethosuximide is blocking the burst because of T-channel blocker or
that's correct.
Blocker, T-channel blocker.
T-channel blocker.
T-channel blocker. Okay.
Yeah.
Thank you.
It's not the most selective one. There's a more selective one now called TTAP2, which also blocks the burst and is also reduced seizure activity.
Okay. Thank you.
Of course. So through because blood glucose is never completely stable in humans, we think that the brain undergoes these either pro or anti-seizure states as a function of how the activation state of AMPK. Okay. So with that, I'm now going to turn our attention to in our last few minutes to how hyperventilation can provoke these seizures. And this is a fairly new project that we're really quite excited about. And the reason why is because, again, it's been known for now almost 100 years that hyperventilation can provoke seizures in children with absence epilepsy. And what is remarkable about this phenomenon is that it does so in almost every single absence epilepsy patient. And the reason why I think that's remarkable is because the genetic etiology that the patients have is likely quite diverse. And yet hyperventilation can somehow transcend that to provoke a seizure in all of these patients. And this is an example. There are a few studies out there now that have quantified this. This is just the breakdown of the patients that were involved. This was a study of 62 patients. And in 100% of these 62 patients, hyperventilation was able to provoke a seizure. And they just described the latency from the onset of hyperventilation to the first seizure occurrence. And it roughly occurs at about 50 seconds. So again, this is a well appreciated clinical problem, but we don't have any idea how this occurs mechanistically. And we think that if we can understand that, especially because it happens in all patients, we might be able to resolve some fundamental sort of properties of absence seizure circuits. So how does this occur? And I should point out, this is a project that was carried out by a very talented graduate student, Katie Salvati, and she was very interested in doing this. And I shared her enthusiasm. However, I told her that I'm not very interested in this project unless you can show me that you can do this in a rat, because if we don't have a rat, we can't really study it, right? So she took that challenge and she developed this following approach. So this is a plethysmography.
Sorry, man. I have a question.
Sure.
In these patients, in the previous slide, were these patients actually showing absence seizures background?
Yes.
All of them were with this absence seizures background.
Yes.
I mean,
so it's a good point. So this only happens in patients with absence epilepsy.
Yeah. Okay. Yeah.
If you do this in a healthy patient, you can, if the age is appropriate, also in young children, you can evoke a phenomenon called hyperventilation-induced high amplitude rhythmic slowing, also known as HIHARS, which looks on the EEG a lot like sleep, but it's not a seizure.
Okay. Yeah.
Yeah. Right. So it only happens so that's why clinically, this is a very powerful tool, because if you have a child that you suspect has seizures, has absence epilepsy, as a neurologist, you can just ask them to hyperventilate and you can have that diagnostic conclusion very quickly.
Sure. Okay. Yeah. Thank you.
Yeah. So how did Katie do this? So she took advantage of this contraption, which is the plethysmography chamber. So this is a sealed chamber where you can deliver different gases, and we retrofitted it so that we could also record EEG. And the first set of experiments I'm going to show you induce hyperventilation by changing the gas composition in the chamber. So here is an example of an experiment. So we're recording the rat. We can change the oxygen composition to a lower value, 10%. And this is a well-known, reliable instigator of hyperventilation. And just as in the humans, when the rat is hyperventilating, you get these occurrence of spike wave seizures. And you can see this as harmonics in this spectrogram in expanded view here. And this is the quantification. This is the same figure I just showed you. So what we do is we generate a peristimulus time histogram that we align to the onset of the gas exchange. And just as I showed you before in stacked histogram format, you can see that within a few minutes of hypoxia, you get almost a twofold increase in the baseline seizure count, as is quantified here. And we also take blood gas measurements, and you can see that as expected, the pH of the blood becomes more alkaline. So this is the pH. It comes up to about 7.6 during hypoxia. And this is the result, we believe, because it's a drop in CO2, and dissolved CO2 is an acidic compound. So the idea is that when you're hyperventilating, you're blowing off CO2, this acidic molecule. And as a result of the removal of that acidic molecule, you're making the blood more alkaline. This is a phenomenon known as respiratory alkalosis. So we wanted to test whether it was indeed alkalosis. So we did the following experiment where we also, in addition to making the chamber hypoxic, we interleave experiments where we introduce hypoxia with CO2, again, an acidic molecule. So this is what the stacked histogram looks like. So we have hypoxia. Again, in this set of animals, it increased the seizure count. However, if we include CO2 in the same animals, we get no effect. So this suggested that, and as quantified here, and this suggested that it was indeed this alkalosis, this respiratory alkalosis, that is driving this increase in seizures. And we also measured blood gas to confirm that the pH was dropping as a function of the combined gas introduction. It actually becomes a little bit more acidic than normal. And we also measured the arterial, the partial pressure of CO2 in the arteries. And not surprisingly, if you include CO2 with hypoxia, it normalizes that property. This is during hypoxia. We also showed that CO2 alone, so just in normoxia without the introduction of hypoxia, can also significantly reduce the occurrence of these seizures, shown in stacked histogram format and quantified here. And again, because CO2 is an acidic molecule, we see this small drop in pH, significant but small, and we think it's sufficient to acidify the blood and make the seizures less pronounced. And this is the quantification of the CO2. Now, one potential concern about this experiment, of course, is that hypoxic activation of hyperventilation is not that normal. It's not what the kids are doing. We're not putting them in a hypoxic chamber. So we next took advantage of an approach that is performed by Patrice Guillenet's lab down the hall. And he can optogenetically modulate the activity of brainstem centers to increase ventilation. So here is an example where Patrice silenced these brainstem centers. And you'll see when the light turns green, he's silencing those centers and the animal immediately stops breathing. It's a pretty impressive response. This is with an inhibitory channel rhodopsin. With an excitatory channel rhodopsin, the animal breathes much faster. So now we can induce hyperventilation in normoxia under normal conditions. And this is the experimental protocol. We do put the animal in this plethysmography chamber, and then we interleave experiments where we have the activation of these brainstem centers in normoxia, and also the activation of these brainstem centers in normoxia plus CO2. And this is an example of a spike wave seizure that's evoked optogenetically by activating the brainstem centers. And this is the quantification in these animals. So in normoxia, we see this pronounced jump in seizures with the laser. And this effect is blunted if you also introduce CO2, just as we saw in hypoxia. So this is I would say, a more powerful example wherein wherein hyperventilation under normal conditions, just as humans would do, can provoke spike wave seizures in animals that already have a spontaneous seizure rate. So this led us to the big question of where is this happening? How is this happening? So the first experiment that we did was to put animals in a hypoxic chamber and just measure neuronal activity indirectly using a CFOS marker. So we put the animals in for about a half an hour. We sacrificed them 90 minutes later. And what we were surprised to find is that we get a lot of activity along the midline of the brain in these so-called intralaminar thalamic nuclei, one of which is shown here, the central median nucleus. The VB is a first order primary sensory thalamic nucleus. And this is just taken from 4 animals, showing you that this response is stereotyped. So we get increased CFOS activity following hypoxia. I'm not going to show you this, but introduction of CO2 with hypoxia reverts it back to the normoxic conditions. And this is quantified here. So the model that we are trying to pursue, and this is admittedly just CFOS activity. I'm just going to show you experiments that are undergoing. But again, what we're highlighting here are the first order thalamic nuclei, the reticular thalamus that I described earlier, and these are the midline thalamic nuclei that almost 70 years ago were implicated in the generation of spike wave seizures, but have largely been ignored in the last 20 or 30 years. So this is, again, where we find FOS activity. So the model that we are proposing is that through hyperventilation, you activate midline thalamic structures that then recruit this canonical circuit that I described. Perhaps we think that midline activity evoked by hyperventilation triggers these seizures by recruiting that cortical burst of hypersynchrony that then generates that rhythmic activity within the thalamic circuit.
Excuse me, but
yeah.
Then you need a connection between the midbrain, the respiratory nuclei,
yes,
reticular formation, to the
yeah.
midline thalamus, and of course there is there, right?
Exactly. So it does exist. So Stereod many years ago showed that there is direct monosynaptic excitation of the midline nuclei from the brainstem, from the reticular structure. So I think there's precedence for it there, but we want to certainly pursue that in more detail. But I'll actually show you a little bit of data that gets to that. I do want to just show you a little bit of, because as an electrophysiologist, I am generally a little bit suspicious of FOS activity. So we have translated this to a mouse model that also has spontaneous seizures. Oops, that also has spontaneous seizures. And just like in the rat, hypoxia can increase the seizure count. And with this mouse model, we are now going to record with silicon probes, hopefully, the activity in those midline nuclei so that we can really look at hypoxia evoked activity. So we've developed this very sophisticated nose cone that we can put over the mouse's nose to deliver different gas compositions. And some preliminary data show that this is actually working, and we can record not only multi-unit activity throughout the cortex and the thalamus, but we can also record single unit activity, which is exciting to us. We haven't yet put this in the midline nucleus because it's hard to get there, but we're going to hopefully do that within the next couple of weeks. So we are going to hopefully directly measure neuronal activity. And then this is just a quantification of some of this activity. With this approach, we can actually see which neurons are turning on first relative to other structures, which I think will be very powerful to try to understand where the seizures are initiating and propagating through the brain. But we're not there yet. So I will show you just in the last few minutes of this talk, I will show you some evidence to suggest to support this hypothesis. So we have cannulated the midline region and we have delivered alkaline solutions. This is a preliminary set of experiments. But in 2 rats, so I'm not showing you the individual rats, but the control is shown here in black. And then if we deliver a pH of 8, which is understandably very high, we can increase the number of seizures that we record. And also, we have found that these midline neurons in the thalamus are pH sensitive. And we've done that both electrophysiologically with our recording pipettes, as well as using imaging approaches. So if we record with pipettes, we get this in the intralaminar nuclei, we get this big inward current during alkaline conditions. We also see a drop in input resistance, suggesting that channels are opening. And what's interesting and speaks to this excitatory input, during alkaline conditions, we also see a barrage of EPSCs. And these are blunted by both TTX and kyurinic acid. I'm not going to show you here. But what I will show you in panel E is we've probed this sort of protocol in other structures, the first order thalamic nuclei, as well as somatosensory nucleus. And it appears that the intralaminar nuclei have this heightened pH sensitivity. So that's what we're trying to track down. We've also done this with calcium imaging recordings. If we introduce alkaline conditions, we can see an increase in fluorescence in these neurons. And this is done now in 2 animals. Okay, so the model then is that, that hyperventilation recruits pH sensitive neurons in the midline thalamus that then somehow recruit this canonical circuit to produce spike wave seizures. And we have obviously a long way to go before we can really connect those dots, but that's the model that we are proposing. And that's summarized here, again, using hyperventilation. We're hoping that we're tapping into some very fundamental core circuitry that drives these absent seizures. And we've shown that hypoxia or optogenetically activating hyperventilation can provoke these seizures in a CO2 dependent way, suggesting that it's pH that's driving this. And we've also found pH sensitivity in the midline that is suggestive that hyperventilation is somehow recruiting these neurons to provoke these seizures. And again, what's really exciting for us is that it does this in all patients, apparently, or at least hyperventilation provokes seizures in all patients, and it also now does it in rats. So it really seems to speak to some fundamental seizure generating circuit. Okay, so with that, I'm sorry I went a little bit over. I just want to thank everyone for their time, and also thank all of the folks that really did the work. Katie Salvati has been instrumental in both projects that I described, both the glucose and the hyperventilation project. Matt Ricker has also provided some great data for both sets of experiments. Adam Liu, as well as older graduate students that have gone off to better and greener pastures, Peter, Lisa, Pasha, and Daniel. So with that, thank you for your time, and I'm happy to take any questions. And I'm sorry I went a little bit over.
Thank you very much, Mark. No, that's okay. You were a very interesting job. So I think we have some questions from the audience. So Liz, do you have a question?
Yes, I do. Thank you for your presentation. It was very interesting. I have a question regarding the formation of ketone bodies and how capable are they to lower significantly the pH within the circuits that you are studying? And do you think that those ketone bodies might be involved in this seizure formation?
It's a really good question, and I don't have an answer for that, but I can tell you that Katie and I have often discussed that at some point, we suspect that this glucose project and this hyperventilation project are going to converge at some point. We just don't know how yet, but it's a really interesting idea that we've considered but haven't tested yet.
Well, thank you.
Yeah.
Any other question from the audience?
Yo, Juan.
Sure, go ahead, please. Thank you, Maria.
Yes, me, Juan Lourdes.
Yes, please, Lourdes.
Very nice talk, very interesting. Thank you, Mark.
Thank you.
And I want to ask you something. We also work in the hypoglycemia model in rat induced by insulin, but we don't study anything that has to do with epilepsy. But when we do these experiments and we inject insulin, we let them rats come into the coma state, into the hypoglycemic coma state. And then we rescue them with glucose. And sometimes we can see seizures because we are monitoring the EEG all the time. We can see seizures after glucose infusion, especially when glucose goes a little bit higher than it should, or we do it a little bit faster. Do you know which is the mechanisms of seizure induction related to this glucose infusion?
I don't. I guess one question that I would have first is, do you know what kind of seizures they are?
They are not absence seizures, but
Yeah.
we haven't studied them in detail, so I cannot tell you.
Yeah.
We just see that the activity is increased. We can see the spikes because we are monitoring the EEG. Mm-hmm.
Yeah, I mean, I can only speculate, and I guess I should start out by saying that, you know, seizure generating circuits and their mechanisms are really quite distinct and diverse across the set of epilepsies. So one of the interesting features of absence epilepsy that really distinguishes it from other forms of epilepsy is that it's one of the few epilepsies where if you increase inhibition, for example, by increasing GABA B receptor activity, you actually aggravate seizures. In a lot of seizure, in a lot of epilepsy forms, increasing inhibition actually makes the seizures less severe. So it's possible that a similar mechanism, whereas somehow decreasing inhibition through a hyperglycemic insult could evoke seizures. It seems plausible, for sure. Yeah.
Okay.
It's a
Thank
good
you.
question. Mm-hmm.
Any other question?
Yes.
Hi, Susana.
Hi.
Please.
Thank you for your interesting talk. I was wondering, considering your final model, if the activation of AMPK could trigger these seizures, and the people that is medicated with metformin could be in the risk of developing these seizures if they...
You know, I am so happy you asked that question, because we have this crazy observation where if we inject only our seizure-prone animals, we only see this in animals that already have spontaneous seizures.
Exactly.
If we inject metformin, IP, and arguably it's a fairly high dose of metformin, perhaps 1.5 times what you would give clinically, you evoke an incredibly powerful seizure response. So the animals, not only does their absence seizure count increase, but they undergo this phenomenon, which is known as status absence epilepsy, where the animals are continuously in spike wave discharge for about 10 minutes. And then the animals start having convulsive seizures. And then in 95% of our rats, they die. And what is remarkable about the sequence of events is that it only happens in the WAG ridge rats, this model of absence epilepsy. We've done the same in Wistar rats, which are, you know, a similar strain as the WAGs, and we see absolutely nothing. We don't see any seizures. We don't see anything. So to speak to your question, is there cause for clinical concern? I don't really think so, because in our hands, we're using a fairly high dose. And the other thing that I would also note is that the seizures, the population of patients that have these seizures is fairly young. So the age of onset is usually around 5 years old. And then many of the patients will grow out of their seizures by the time they're 18 or 19. And I don't think, even though I've looked often on the internet to try to see, is there this relationship? But I think my guess, and I'm not a diabetes person, is that my guess is that most of the folks that are taking metformin are in a significantly older population. Yeah, but it's a great question. And it let me describe this crazy observation where not only does metformin cause seizures in seizure-prone rats, it's ultimately lethal, which was very surprising to us.
Yeah, it's very surprising. Thank you very much for your talk. It was very nice. You say that you have to use a high dose of metformin, and that's okay. But the problem with physicians is that they mix medications very often.
Yeah.
And adult patient almost always has a polypharmacy. And I have old studies when you give benzodiazepines, for example, for a long time, and then you take them over and they're out, and the animal develops seizures.
Yeah.
Just by taking out the benzodiazepines. If you are trying a patient with these drugs, and they also have metformin, and you are in danger. I mean, you wouldn't need a higher dose of metformin because the synergy between benzodiazepines and metformin could be dangerous. It's just a hypothesis, but these are very useful, of course, clinical drugs, but very often mixed.
Yeah, and in fact, I've seen in the literature some cases where there's a metformin overdose. I'm not really sure the circumstances that lead to that, but it can happen. Yeah.
Actually, I have a couple of questions, Mark. Very, very interesting. The job you have done about this absence epilepsy. There's been for a long time known that this petit mal epilepsy, as originally known, is precisely occurring mostly in young children, and seems to disappear when they get older. Is there any mechanism known about this decreasing in the disappearing actually in the current of these seizures when you are getting older? I mean, is there any mechanism about this?
Any real mechanism? I don't think so. I get this question asked often, and we simply don't know. It does seem to coincide with sort of the end of development of the human brain, which is around in the early 20s, I would say. But what's also interesting is that, as I mentioned in my introduction, there's two forms of absence epilepsy: childhood and juvenile, and they are distinguished largely by their age of onset. So childhood is around 4 or 5, juvenile is maybe 6 or 7. They both have absence seizures. The juvenile absence epilepsy patient does develop other seizure types, but unlike the childhood population, the juvenile population never grows out of their seizures. In fact, they're a clinically difficult population to treat. Their seizures only get worse and worse with age. How the real childhood absence epilepsy patients grow out of their seizures is a great question, and we really don't know. Yeah. I wish we would know, but I think it might have something to do with just the maturation of the brain with time. Yeah.
Yeah, you know, there is also some parallel hypothesis about this regarding to the existence of this channelopathy of T-type calcium channels that is being shown is very common in these people that has absence epilepsy. This channelopathy is some several point mutations in the CAP3.2 channel. And also Perez Reyes and I, we have done some recordings about that and reports showing that the channelopathy of CIV 3.2 channel is kind of a possible likely channelopathy for this absence epilepsy. Do you have any data or information about this relationship with the absence epilepsy?
So I haven't looked at the literature in a while. I mean, for sure, gain of function mutations in T channels have been identified in the absence population. But again, sort of speaking to the polygenic nature of the disease, none of those mutations really account for a significant portion of the population. So it's really, you may be familiar with Dravet syndrome, which is this horrible disease that occurs in young children, and it's also a seizure. It's associated with seizures. So in Dravet syndrome, there is an identified mutation in SCN1A in about 70% of all patients. So it is clear that that is a relevant mutation. And I'm not saying that that T-channel mutations are not irrelevant. They just account for a small population. So we really now view this disorder as a polygenic disease, which multiple mutations have to come together to produce the epilepsy, which of course makes it very difficult to study in the lab, right? Yeah,
that's for sure. Yeah, unfortunately, there is no just one protein involved, one gene.
Yeah, that's right. Yeah, I mean, this is the realization that the epilepsy world has really come to, I think, in the last 5-10 years. In the early 2000s, when there was just this huge push in genetic biology, I think the epilepsy community, as well as many other communities, thought we would identify the mutations and then the cures would come shortly thereafter, right? And it just hasn't. It's proven to be much more complicated than that.
Yeah, sure. That's for sure.
Yeah.
All right. Is there any other question in the audience? Okay. Well, so again, Mark was really very productive. This seminar, very nice, very interesting data. So yeah, as I was telling you at the beginning, it might be one reason to start some potential collaboration between
I would love that
relation. Yeah. So yeah, thank you very much for your seminar. Very interesting, and I hope this could be also very interesting for several labs here in the institute. So thank you very much.
Of course.
And we will continue with our chat lately. You agree? Okay.
Okay. Thank you so much. I really enjoyed it.
Thank you very much, Mark.
Yep.
And thank you all. Thank you all for assisting and waiting until the end.
Yep. Bye-bye.
Have a nice day.
Okay. Thank you.
Have a nice day over there.
Thank you.
Bye.
Bye.